Subscribe this channel by pressing this button for being updated from Biomedical and Healthcare Society.
Hello guys, welcoming you all in another episode of Wikitech Updates. I am Amit and you are watching Techomed. So without any further delay, let's get started. The very first update is about a new cell culture technique for the high-speed cell production. Researchers at Newcastle University in the UK has developed a new culture technique that allows scientists to produce enormous quantities of cells using a very small growth areas. The method is a peptide amphifill coating that allows cells to grow and then peel away and reattach from the growth surface, leaving space for the more cell to grow. The method could be very useful in growing the huge number of cells required for upcoming cell therapies. This allows us to move away for the first time from the batch production of the cells to an unready-made processes. This is fantastically high number of patients in the cell in need of the cell therapy, such as those suffering from heart and cardiac, skin cancers related diseases, and also on those this technology provides a much needed solution while saving costs, reducing materials, and improving the quality and the sterilization of the final product. The other update is about the use of low energy X-rays on the cancer cells. Sensus Healthcare is a medical device company that focuses on providing non-invasive and cost-effective treatment non-melanoma, skin cancers, and keloids utilization superficial radiation technology, that is SRT. Their FDA cleared SRT technology is used to effectively and safely treat oncological and non-oncological skin conditions. The radiation is focused onto cancer cells almost exclusively and it penetrates no more than 5 mm under the surface of the skin. Sparing nearby tissues, superficial radiation therapy, that is SRT, is a low energy photon radiotherapy technology that delivers precise, calibrated, low dose radiation that effectively destroys basal cell carcinoma and squamous cells, carcinoma as well as the non-malignant tumor cells that causes keloids. Proprietary sensors designed and made applicators, the SRT-100 and SRT-100 Vision, allow doctors to focus 100% the low energy photon radiotherapy directly on the cancer cells, penetrating no deeper than 5 mm below the surface, allowing for the preservation of the surrounding tissues. The another update is about weighing the cancer cells for their susceptibility in the chemo. Researchers at MIT have developed a new method to measure how well multiple myeloma patients will respond to a specific chemo agent or combination of them. This process involves using an incredibly sensitive apparatus that can weigh individual cancer cells. If the cancer cells reduce the rate at which they gain mass when exposed to a chemo drug, then they are susceptible to it, and so to be a good fit for using as therapy. This technique can be used to test the susceptibility against a variety of chemo drugs from just one batch and could pave the way to personalized cancer medicine. The idea is simple in the principle but require more very sensitive equipment that can weigh something as small as a cancer cell. The technique requires very few cells, so it is possible to test a wide variety of drugs and their combinations on a sample for just one biopsy. There is a lot of things to be addressed regarding this. The other updates is about the quantum dots, which lights up the tumors in tachycolors. Quantum dots are tiny nanoparticles which made of semiconductors that have unusual optical properties. In medicine, they may be very useful because they emit light when stimulated by electricity or an external light source, making them easy to spot in diagnosis tests. A major challenge of putting quantum dots into clinical practice is how to attach them to antibodies that seeks out disease without ruining the functionality of neither the quantum dots nor the antibodies. Researchers at the Riken Research Institute in Japan have now developed what seems like a foolproof way to blind antibodies with quantum dots. To validate the new capability in practice, the researchers used attached quantum dots to antibodies that seeks out the HER2 receptor commonly found on many breast cancer tumors. The result was that they were able to light up the tumor samples while keeping in the non-concerning cells in the dark. The other update is about the AG Scan Go, which is an Artificial Intelligent Malaria Parasite Counter. EasyScan-Go is an artificially intelligent microscope developed by Chinese manufacturer Motic. Has the capability to automatically and accurately quantify malaria parasites in a blood sample. Utilizing machine learning algorithms, the microscope is so effective that it can identify the amount of parasites present in under 20 minutes. EasyScan-Go works through a calibration of the digital slide, scanning and the diagnostic software module based on the machine learning and neural network. The machine was trained by feeding it thousands of blood smell slides, annotating by experts. Malaria affects 200 million people a year and 40 lakh of those cases are fatal. Typically found in tropical and subtropical countries, strains of the malaria are becoming resistant to the more effective drugs. Though physicians can do the rapid diagnosis tests at their disposal, physicians and researchers depend on the microscopes like the EasyScan-Go to quantify the amount of strain of malaria present in the sample. EasyScan-Go makes monitoring drug effectiveness more efficient, freeing up the expert microscopics as well as making the ability to quantify malarial strains available to the wider medical community. So that's all about the Wiki Tech updates from Biomedical and Healthcare Society. If you have any query or suggestion, you can give it in the comment section. Don't forget to subscribe this channel. If you like this video, hit the like button and share this video with others. So this is Amit Mohan Raman signing out. Jai Hind.